is Anna Birnbaum, and today I will be presenting my master's research titled Removal of Nitrogen and Phosphorus from Nursery Runoff using Zeolite and Woodchips. Okay, first I'll go over an outline of what I will be presenting today. First I'll go over an introduction with background information, followed by my objectives. Next I'll go over my experimental design, followed by the results, and finally the conclusion. Okay, introduction. First I'll go over some introductory information about the nursery industry. The United States nursery industry has about $4.3 billion in sales annually, and they are the largest users of fertilizers per unit area in the agricultural sector. And due to this high use of fertilizers, there is a high concentration of nitrogen and phosphorus in the runoff leaving the nurseries. This nitrogen and phosphorus-rich water can lead to eutrophication, or the formation of algae blooms. This can occur in waterways downstream from the nursery, in lakes and rivers, or also on-site in the nursery retention ponds. These algae blooms lead to a depletion of oxygen and also dead zones. If the algae bloom occurs on the nursery retention pond, the grower would need to treat the water more before they can recirculate that water back onto their crops. Okay, eutrophication has led to the implementation of best management practices. Some of this has come out of regulations, but also nursery growers doing this on their own to treat the water before it goes into the retention ponds. Best management practices can be in the form of irrigation, as in using spray stakes instead of overhead irrigation, fertilizer use, using controlled release fertilizers instead of overhead fertigation, and also through nutrient and water capture techniques. This is the type of best management practice we created in this experiment. We wanted to capture the water at a retention pond and then run that water through a series of reactors to remove the nutrients. Okay. Now I'll go over the different components we use to remove the nitrogen and phosphorus from our wastewater. First, woodchip bioreactors. Woodchip bioreactors are a commonly used best management practice in the Midwest in corn and soybean production. They use the biological process of denitrification, which is the conversion of aqueous nitrate into harmless dinitrogen gas. They are low cost, as they are just a large ditch in the ground filled with wood chips, and long lasting. They have been shown to last up to 10 years. Okay, next, phosphorus removal. Phosphorus removal in conventional wastewater treatment can be done biologically. In this process, the wastewater is run through a series of anaerobic and aerobic tanks filled with polyphosphate accumulating microorganisms. These microorganisms remove the phosphorus from the wastewater, and this would be difficult for us to do out in the field in a nursery setting in order to create these anaerobic and aerobic alternating tanks. Okay, so then chemical precipitation is the other form of phosphorus removal in conventional wastewater treatment plants. Chemical precipitation involves injecting a chemical coagulant into the water treatment chain. These are usually in the form of ferric salts. The ferric salts bind with the phosphates to form precipitates, which then can be removed from the wastewater as a sludge. Commonly used salts are ferric chloride and ferric sulfate. In this experiment, we use ferric sulfate. Okay, third, zeolite. Zeolite is a aluminosilicate clay mineral. They have a high cation exchange capacity, meaning they have the ability to easily exchange cations such as iron and ammonium. Even though they are able to exchange cations easily, they have a very low affinity for anions. This has led to the creation of surface modified zeolites. Surface modified zeolites thus far have only been tested in a lab environment. Oftentimes they coat the zeolites in iron to form a cation bridge to give the zeolite positive charge for the anions to bind to. In this experiment, we used the ferric sulfate, hoping to bind the phosphates to the zeolite. To illustrate this, here is the negatively charged zeolite, and then we bind the iron to the zeolite, and that allows the phosphates to bind to the zeolite using the iron as a cation bridge. Okay, now to my objective statement. My objective is to examine the efficacy of ferric-aided zeolite-filled molecular sieves in conjunction with or without preceding woodchip bioreactors for removal of nitrogen and phosphorus from horticultural nursery runoff on a pilot scale as a potential BMP for horticulture nurseries. Or more simply, my objective was to remove nitrogen and phosphorus to determine the impact of zeolite and wood chips on water chemistry, to remove excess injected iron, and in the future, we did not evaluate this in this experiment, but we suspect that the nitrogen and phosphorus laden zeolite that is created in this process can be used as a value-added substrate amendment for the nursery industry. Okay, now to my material and methods. The photo on your left is a picture of the nursery runoff pad we created. Water from this runoff pad, well, the plants on the runoff pad were fertilized with controlled release fertilizer, and then daily they are irrigated with fertilizer-rich water by fertigation. This water ran off into the runoff pad, or the retention pond on the right. And then the three raw materials used in the experiment was zeolite on the left, wood chips in the center, and then on the right is an image of the ferric sulfate injection point. Okay, now here is a diagram of the layout of my experiment. It was a completely randomized design. I'll go into this image in more detail. So water from the retention time would be pumped from the retention pond, was pumped up to the reactors at a rate of 15,000 liters a day continuously for 12 weeks. At the same time, ferric sulfate was continuously injected into the system at a rate of 4 parts per million. Note that before the start of the 12 weeks, we ran pond water intermittently from the retention pond for about a month in order to colonize the woodchip bioreactors with native microorganisms. In the image here, you can see the systems. Okay, here, the image on the left shows the water flowing into the bioreactors. And then the image on the right shows the water, the outflow point. Water flew at a continual 800 mils a minute for the 12 weeks. There was some difficulty maintaining the rate of flow at 800 mils a minute, required daily calibration. Okay, it has 3 treatments. There's treatment Z, zeolite treatment. Treatment C is the water blink control. And treatment WZ is 4 woodchip tanks followed by a zeolite tank as shown in this image. There are 3 reps of each treatment. Okay, and now this is showing treatment Z, which is just 1 zeolite tank filled with 8 cubic feet of zeolite. The woodchip tanks were filled with 8 cubic feet of woodchips. And then treatment C is the water blink control, just 100-gallon tank filled with water. Okay, in order to allow water to flow easily out of the zeolite tanks at the rate of 800 mils a minute, and for water to flow between the woodchip tanks at a rate of 800 mils a minute, we had to install drainage filters. The zeolite tank was a perforated pipe covered in mesh, and then for the woodchip tanks, we used perforated PVC pipe. And here are just some more images of the water flowing into the systems. On the left, you see a zeolite tank. Okay, and now here is an aerial image of the experimental layout. I'll go over this in more detail. Electrochemical monitoring. I monitored electrochemical properties weekly for EC, pH, and oxidation, temperature and oxidation reduction potential. Used a YSI meter. You see on the right, I had to create a sampling well in the zeolite tank in order to give the probe somewhere to lay as I took the measurements. Okay, and that back to the aerial image. I took, this is showing the sampling points for the YSI meter for the electrochemical properties. I sampled in the center of each of the woodchip tanks, and then at the zeolite tank in the center following the woodchip tanks. In the same, I sampled in the center of the zeolite tanks and the control tanks, 8 centimeters deep below the water surface. Okay, nutrient monitoring. I monitor nutrients weekly for total dissolved iron and phosphorus, and then also for total dissolved inorganic N, also abbreviated as DIN, D-I-N. This is a combination of ammonium N, nitrite N, and nitrate N. And this is showing the sampling points for the nutrient samples. For the woodchip zeolite treatment, the nutrient samples were collected weekly in front of the first woodchip tank at the inflow pipe, after the 4th woodchip tank, and then following the zeolite tank, following the woodchip tanks. And then for the zeolite and control treatments, nutrient samples were taken before the tanks at the inflow pipe and then at the outflow pipe. We also collected zeolite and woodchip samples for nutrient analysis. These samples were collected on week 1 and week 12 of the experiment. Okay, and now onto the results. pH did not vary significantly for the control or the zeolite treatment over the course of the experiment. However, the woodchip zeolite tank did have an average increased pH of 1 pH point over the control and the zeolite tanks. Okay, oxidation reduction potential. The control and the zeolite tank did not vary significantly from each other or from the pond over the course of the experiment. However, the Woodchip zeolite treatment WZ did have a significantly reduced oxidation reduction potential of negative 220 millivolts. Temperature. Temperature, again, did not vary significantly between the control and the zeolite treatment. However, temperature was on average 1 degree Celsius warmer for the Woodchip zeolite treatment. Okay, now onto the nutrient results. On all my graphs on the x-axis, WAI refers to weeks after initiation. On this one, y-axis is total percent phosphorus removed or reduced. Phosphorus removal did not vary significantly between the control and the Woodchip zeolite treatment. However, there was significant removal for the zeolite treatment with an average of 27% weekly mean removal. Okay. And here are the same still phosphorus removal data, but in terms of cumulative flux. See, there is no significant phosphorus removal for the control or the Woodchip zeolite treatments. However, there was a total of 124 grams of phosphorus removed from the zeolite treatment. So we had designed this experiment to remove nitrogen and phosphorus using the Woodchip zeolite treatment. However, there was not significant removal of phosphorus in the Woodchip zeolite treatment. So why did we not see significant removal? One explanation could be that, well, from previous research, we know that phosphate absorption decreases as pH increases. The pH of the Woodchip zeolite treatment was 1 pH point higher than the other two treatments. At ultra low or ORPs, less than negative 150 millivolts, phosphorus has been shown to desorb from zeolite. We also suspect we had the formation of coke. Coke is a highly carbonaceous carbon species that can form in highly reduced sulfur-rich environments. We had both of those. The coke is in the photos on the right. You see the zeolite went from its normal creamy white color to this dark black color. The coke coats the pores of the zeolite, blocking the binding sites for the ions. Also, we suspect that the wood chips may have leached phosphorus, as they have an inherent phosphorus concentration of 450 milligrams per kilogram. This is why we placed the zeolite reactor after the wood chips, suspecting that the wood chips would release phosphorus. However, due to these highly reduced conditions and the coke formation, the zeolite was not able to bind that phosphorus. Also, from our material analysis, we found that this reduced phosphorus as compared to the oxidized the oxidized zeolite had 3 times as much iron absorbed to it as compared to the reduced zeolite. If there's not iron bound to the zeolite, then there was not that cation bridge for the phosphorus to bind to. And finally, the zeolite placement. Again, we designed the system this way, with the zeolite following the wood chips to absorb the phosphorus that may leach from the wood chips, and also to absorb ammonium that may have come from the wood chips. However, due to the highly reduced conditions, the zeolite was not able to work as we had planned. Okay, now this graph here shows weeks after initiation on the x-axis, and then TP cumulative flux again on the y-axis. However, now instead of just the zeolite treatment, which is the purple line, control the blue line, and the woodchip zeolite treatment, the red line, we also have the woodchip zeolite treatment broken down into wood only. So we took a sample before and after the wood tanks, and then zeolite only. So that's from the sampling point post wood tanks, and then post zeolite. So you'll see that we did have phosphorus increase for the zeolite only and the woodchip and zeolite tanks only by the end of the experiment. However, the woodchip tanks were actually removing phosphorus at the end of the experiment. But at this point, the zeolite tank began to release phosphorus. We suspect at this point the coke formation had become so rich that the phosphorus could no longer absorb, and previously absorbed phosphorus began to be released. Okay, and this is showing what I just talked about. You have the sampling points for the woodchip wood only is from the first W before the wood chips, and then post wood chips, and then for the zeolite only in front of the first zeolite, in front of the zeolite tank, and then after the zeolite tank. So zeolite was only was initially removing phosphorus until week 6, and then we suspect the coke formed and began releasing phosphorus, and the wood chips were adding phosphorus until week 7, when they probably had finished leaching all of their inherent phosphorus. Okay, now on to nitrogen. Nitrogen, the control in the zeolite treatment did not have significant removal over the course of the experiment. However, the woodchip zeolite treatment did remove an average of 14.3% of nitrogen per week. Here's the nitrogen data again, but in terms of cumulative removal. You see there's no significant cumulative removal for the control treatment. The woodchip zeolite treatment did remove 465 grams of nitrogen. And then the zeolite treatment actually had an increase in nitrogen. We suspect this is because during the acclimation period before the start of our 12 weeks, from our zeolite analysis, we saw that zeolite only tank had more nitrogen sorbed to the zeolite at week 1 than it did at week 12. So we suspect this increase in total zeolite is due to that initial nitrogen being released as that zeolite became overloaded with iron. Okay, the woodchip zeolite treatment did remove nitrogen. However, it was not as much as we suspected it would. Woodchip bioreactors remove on average 33 to 100% of nitrate in the water, depending on hydraulic retention time. We designed our Woodchip bioreactors for about a 5-hour hydraulic retention time, which should have removed around 60% of the nitrate from previous research. So these graphs, we decided to look at what was causing this. The first graph on the left, see ammonium flux. You see, and then on the right is nitrate flux. Even though we had a great reduction in nitrate, we also had a great increase in ammonium. There was 1,400 grams of nitrate removed and 1,000 grams of ammonium created. So still a net increase or net decrease in nitrogen. But next we had to find out why we had this ammonium creation. So we suspected we would have denitrification, which is the removal of nitrates through the pathway that results in dinitrogen gas. However, instead, we had the conversion of nitrates to ammonium. This is the process of DNRA, dissimilatory nitrate reduction to ammonium. This can occur in woodchip bioreactors that have excessive retention times, which we suspected we had due to the highly reduced conditions. DNRA can also occur in environments with high sulfur concentrations. There are differing theories of why this occurs. Some believe that the denitrification or DNRA bacteria function better in high sulfur environments, and also that sulfur can block the enzyme that drives denitrification. It is also believed that DNRA occurs in environments with nitrate limiting conditions. It's a way for the bacteria to conserve nitrogen by converting it to ammonium instead of into dinitrogen gas where it is lost. Okay, so now that we believe we have excessive retention times, at the end of the 12 weeks, I conducted an quick hydraulic retention time experiment, injected bright green dye into the system, and monitored it as it flew through the reactors for 12 hours. At the end of the 12 hours, there is still dye remaining in tanks 2 through 5 of the woodchip bioreactors, and also bright green pockets of dye in tank number 2. From this, and also the ultra low ORPs, we suspect we have hydraulic retention times greater than 24 hours, resulting in these negative chemical processes such as DNRA. The formation of coke and also the strong rotten egg smell around the bioreactors, and also due to the DNRA, we suspected we had some sort of sulfate reduction going on in the form of hydrogen sulfide gas. So with a gas vent meter, we measured gas emissions at one point over the course of the experiment. Results found that the woodchip zeolite tanks were emitting 16 parts per billion sulfur dioxide. which is a byproduct of hydrogen sulfide, and also that the Woodchip bioreactor tanks were emitting 50% more methane than treatment C and Z. Okay, iron removal. We injected iron into this experiment as a means for removing phosphorus. So we hope to have no iron leaving the experiment. The zeolite tanks did a pretty good job with this. They removed 89% of the iron injected. The control and woodchip zeolite tanks also removed about 60% of this injected iron. So finally, my conclusions. Neither treatment zeolite or woodchip zeolite was effective for removing nitrogen and phosphorus simultaneously. The zeolite treatment had an average phosphorus removal of 27%. We suspect the highly reduced conditions resulted in poor performance of treatment WZ. And in the future, the woodchip zeolite treatment train needs to be properly engineered to avoid stagnation zones or excessive hydraulic retention times for effective remediation of both nitrogen and phosphorus simultaneously. Okay, now my acknowledgements. I'd like to thank my advisory committee, Dr. Jim Owen, Dr. Lori Fox, and Dr. Alex Niamera. Technical assistants, I'd like to thank Julie Brindley, Dr. Matt Ike, and Dr. Sarah White at Clemson University. And to my funders, the USDA Clean Water 3 grant and the Hampton Roads AREC for Virginia Tech.